DNA fingerprinting was invented in 1984 by Professor Sir Alec Jeffreys after he realized you could detect variations in human DNA in the form of mini satellites. A DNA fingerprint is a unique pattern in DNA that differentiates individuals. DNA fingerprinting was first achieved by RFLP analysis, in which DNA is collected and cut into smaller pieces by restriction enzymes, generating DNA fragments of different sizes as a consequence of variations between DNA sequences of different individuals. Then, the fragments are analyzed and separated using gel electrophoresis. Then, the fragments are submitted into a Southern blot procedure, and the results are analyzed. There are several ways a DNA fingerprint can be analyzed. Some of them are PCR analysis, which help because it confines the replication to a specific DNA sequence of interest, STR analysis, which is the short tandem repeats, and AMP-FLP, which is the amplified length polymorphism. DNA fingerprints can be used in criminal and forensic investigations to clarify paternity. Also, it is used for genealogical research or medical research, among others. In this practice, the PCR technique will be used, and the materials are the following: PCR tubes, a centrifuge tubes of 2 milliliters, which are colored, the master mix 2X, primers, CSI DNA samples, suspect A DNA sample, suspect B DNA sample, suspect C DNA sample, suspect D DNA sample, micropipettes, ice, orange G loading dye, CSI allele ladder, a power supply, and electrophoresis materials and equipments.
For the setting of the PCR, the following steps were done. First of all, 5 PCR tubes were labeled as CS, A, B, C, and D. A microtube was prepared adding 30 microliters of master mix and 1.2 microliters of primer, giving a total volume of 31.2 microliters. Then, 5.2 microliters of this solution was added to each tube. Following this, 4.8 microliters of DNA were added to the 5 PCR tubes. For the CS tube, the chromatin DNA was used. For the rest of the tubes, DNA from suspects A, B, C, and D were added respectively. Then, the samples were placed in the thermocycler with the following configuration. Next, for the electrophoresis of the samples, 1% agarose gel was prepared. After this, 10 microliters of orange loading dye was added to each of the PCR tubes. Later, 15 microliters of the DNA marker and 50 microliters of each sample were loaded into separate wells in the gel. Then, the electrophoresis was performed at 100 volts for 30 minutes. Finally, the gel was placed in the transilluminator to see the results.
For the last 30 years, the development and application of genetics has revolutionized forensic sciences. In 1984, the analysis of polyformic regions of DNA produced what was termed a DNA fingerprint. In 1986, appeared the first case where DNA fingerprint was used in order to convict a murderer. Since then, the use of genetics was rapidly adopted by the forensic community and plays an important role worldwide in the investigation of the crime. Both the scope and scale of DNA analysis in forensic science is set to continue expanding for the foreseeable future.
To begin with the following discussion, a list of crucial concepts will be explained. First, a genotype is a person's own genetic makeup. Usually, it is considered as a snapshot of genetic information from different loci across the genome. On the other hand, a locus is a particular location in the genome or in a chromosome. Meanwhile, an allele is a particular variation of the individual locus. It is important to mention that non-coding DNA is used for forensic analysis because it does not carry any information about gene expression patterns or person's biological state.
The way in which the forensic laboratories analyze the material recovered from crime scene are pretty straightforward. As shown in the diagram, the sample should be first recovered and then they enter into the forensic laboratory and a forensic genetist has to compare the victim's sample with the suspect's. Several stages are involved with the analysis of genetic evidence. First of all, it's necessary to identify or even characterize the material used. Then, a DNA extraction and quantification is needed. After that, the PCR amplification will let the genetist to compare the material in study with the positive control, finally giving the results to the appropriate authorities.
For this laboratory practice, samples obtained from professor were used. These samples were labeled as A, B, and C and D. The design of experiment included a crime scene sample, which was the one to be compared. In total, 5 PCR tubes were used. For the PCR reaction, a pair of primers were used in order to define the region of the DNA samples that were analyzed. As Goodwin and collaborators state, when designing primers for forensic analysis, it is important that they will bind to conserved regions of DNA and therefore actively differentiate the samples from a person to another.
The results obtained after the electrophoresis of the PCR product were not the best case scenario for the four teams. In wild gel, it did not appear any band, and in our gel, only the CS sample was shown. Two possible things can justify this. The first one is the absence of DNA at the moment of the loading the gel. This might be due to a pipetting error, either before preparing the PCR reaction or before loading into the gel electrophoresis. And the second one is the wrong annealing of the primers to the DNA template, but this can be discarded as the CS sample actually amplified, discarding the conclusion of wrong annealing of the primer because that the sample actually amplified.
On the other hand, the DNA fingerprinting analysis that was done by Quorum in 2018 using the same reagents gave positive results in detection of the suspect. As Goyal defined DNA fingerprinting, it is a method of genetic discrimination because, in order to get the results, it is necessary to compare the positive control, in this case the crime scene sample, with the rest of the samples. For this experiment, lane 3 corresponds to the crime sample that has an evident match with lane 10, giving the result of the correlation between them. The rest of the bands did not show the same pattern as the crime scene samples, resulting in a negative relationship with lane 3.
Before finalizing this report, the answers of an exercise in the laboratory handout about the BXP007 that was provided by Victor Rodriguez to support the previous knowledge registered in this discussion are mentioned, making the assumption that if the BXP007 locus is found exactly the same frequency in a population and there are 8 possible alleles at this locus, the frequency of one allele from this locus would be 1/8. Also, the law of independent assortment states that allele frequency can be multiplied together to give a resulting genotype frequency at a single locus. So, each allele frequency is multiplied together to give the genotype frequency 1/8 times 1/8 equals 1/16. Finally, if a pool of 13 suspects and only one suspect had a genotype that matched the BXP007 locus found in a crime scene, we would be satisfied that we found the perpetrator, because we know that 1 out of 64 individuals are likely to have the same genotype as the suspect, and we are only examining 13 suspects.
In conclusion, we can say that this practice was not a proper one because of the final results. Improvement in the pivoting skills have to be done in order to achieve the effective and expected result.
DNA fingerprinting is important nowadays because it can physically connect a piece of evidence to a person or rule out someone as a suspect. It can also show you who your parents, siblings, and other relatives may be. And it also helps to identify dead bodies that's too old or damaged to be recognizable. DNA fingerprinting is extremely accurate, and most countries now keep DNA records on file in much the same way police keep copies of actual fingerprints. In the medical terms, it can match tissues of organ donors with those of people who need transplants, and it helps to identify diseases that are passed down through your family. It also helps to find cures for other diseases called hereditary conditions.